Hello friends, welcome to my channel Helping Hand for Nurses. This video includes important points from the subject microbiology which will be helpful for you in preparing most of the nursing exams. If you want the PDF, I will provide you in the description box. So please watch the video, and if you like this video, please like and subscribe it. Let us start the video. First, we can see some of the major milestones and major discoveries in microbiology. So usually the important points that are describing in this slide have been asked in most of the previous nursing exams. So first one, doctrine of spontaneous generation was proposed by Aristotle. That is, a theory of spontaneous generation was proposed by Aristotle. Theory of spontaneous generation means that is a life arose from non-living matter. So this was proposed by Aristotle. Cell was discovered by Robert Hooke in 1665. Cell was discovered by Robert Hooke in 1665, and the cell theory was proposed by Schwann and Schleiden. Cell theory was proposed by Schwann and Schleiden. First microbes were observed by Antony van Leeuwenhoek, and he is known as the father of microbiology. He observed RBC and protozoa through a microscope. So Antony van Leeuwenhoek is the first person who observed microbes using microscope, and he is known as the father of microbiology. Next person is Joseph Lister. He started to use antiseptics while performing surgeries. Joseph Lister started to use antiseptics while performing surgeries, and the first used antiseptic was carbolic acid, and the other name for the carbolic acid is phenol. Golden age of microbiology is between 1857 to 1914. So rapid advancements and discoveries occurs during this period, which will lead to the establishment of microbiology as a science. So this period is known as the golden age of microbiology, that is from 1857 to 1914. Microbiology established as a science by Louis Pasteur. Louis Pasteur is the father of modern microbiology. So father of modern microbiology is Louis Pasteur, and father of microbiology is Antony van Leeuwenhoek. Next, we can see some of the contributions given by Louis Pasteur in the field of microbiology. So father of modern microbiology is Louis Pasteur. Father of modern microbiology is Louis Pasteur. And Louis Pasteur disproved spontaneous generation. Louis Pasteur disproved the theory spontaneous generation, and he developed germ theory of disease. And he has given the scientific basis for fermentation. Also, he introduced methods of sterilization like sterilizer, hot air oven, and autoclaving. So, Louis Pasteur introduced the methods of sterilization like sterilizer, hot air oven, and autoclaving. Developed pasteurization. He founded that the life can be exist without oxygen. Louis Pasteur coined the term vaccine. He discovered vaccines for rabies, chickenpox, cholera, and anthrax. And he used swan-necked flask in his experiment. So Louis Pasteur is the father of modern microbiology, and he coined the term vaccine. He discovered a vaccine for rabies, chickenpox, cholera, and anthrax, and he developed a germ theory of disease and also developed a pasteurization. Next important point is that Louis Pasteur is a person who developed a germ theory of disease, but the germ theory of disease was proved by Robert Koch. So germ theory of disease was proved by Robert Koch, but it was developed by Louis Pasteur. Next, we have some of the contributions given by Robert Koch in the field of microbiology. So he discovered simple staining techniques. Robert Koch discovered simple staining techniques, and he developed techniques for estimating colony forming unit per ml. And he used the Petri dishes, and also he has developed pure culture technique, and he discovered TB, anthrax, and cholera microbes. He discovered tuberculosis, anthrax, and cholera microbes, and he formulated Koch postulates. He formulated Koch postulate. What is Koch postulates? First point is that is first criteria is the microorganism must be found in deceased but not in healthy individuals. That is, a microbe should be found in the deceased person but it should not be found in healthy individuals. And the microorganism must be cultured from the deceased individual. We should be able to isolate the microorganism from the deceased individual. Inoculation of healthy individual with a cultured microorganism must recapitulate the disease. So if you are inoculating the cultured microorganism into healthy individual, it should produce the disease in the healthy individual. And the microorganism must be re-isolated from the inoculated diseased individual and matched to the original microorganism. And the fourth criteria is that the microorganism should be isolated or re-isolated from the deceased individual and should be matched to the original microorganism. So in simple sentence, first criteria is that the agent is observed in all cases of disease. That is, agent should be or microbes should be observed in all cases of disease and should be absent in healthy organisms, and the microbes is able to be isolated in a pure culture, and when it is introduced into a healthy host, the organism experiences the same course of disease. That the microbes should produce the same course of disease in the healthy individual, and the agent can be re-isolated in the pure culture. So these are the 4 Koch postulates. And the limitation of this Koch postulate is that Streptococcus pyogenes produce several diseases, and HIV can only be seen in humans, and Mycobacter leprae and Treponema pallidum cannot grow in culture media. So these are the limitations in this Koch postulates. Next we can see the major discoveries in the field of microbiology. Robert Koch discovered the causative agents of anthrax, tuberculosis, and cholera. The causative agent of anthrax is Bacillus anthracis, tuberculosis Mycobacterium tuberculosis, and cholera is Vibrio cholerae. And Robert Koch proved the germ theory of disease, and this germ theory of disease was developed by Louis Pasteur. Louis Pasteur is known as the father of bacteriology, and he developed fermentation and he disproved the theory of spontaneous generation, developed pasteurization, and developed immunization technique. Ronald Ross found out that the malaria spreads by mosquitoes. So Ronald Ross discovered that the malaria is usually spread by mosquitoes, and the Laveran found out that the causative agent of the malaria is Plasmodium species. Edwin Klebs identified or discovered the causative agent of diphtheria. That is, Edwin Klebs identified or discovered diphtheria bacillus, and Loeffler cultivated the causative agent of diphtheria. So the causative agent of diphtheria is Corynebacterium diphtheria, and it is also known as the Klebs-Loeffler bacillus. Theodor Eschrich discovered the causative agent of travelers diarrhea, which is E. coli. Theodor Eschrich discovered E. coli, which is the causative agent of travelers diarrhea. David Bruce discovered the causative agent of Anduran fever or brucellosis, and the causative agent is Brucella melitensis. Dmitry Ivanovsky and Martins Benjamin discovered the causative agent of tobacco mosaic disease, and the causative agent is the Tobamo virus. So Dmitry Ivanovsky discovered the first virus. Martins Benjamin is known as the father of virology. Joseph Lister introduced aseptic surgery. Christian Graham introduced the Gram staining procedure. Van Bering introduced diphtheria antitoxin. Paul Ehrlich proposed the theory of immunity, and he introduced the first synthetic agent against syphilis, that is salvarsan or asfenamine. Alexander Fleming introduced the first antibiotic, that is penicillin. Hansen discovered lepra bacillus. Neisser discovered gonococcus. Frankel discovered pneumococcus. Weisselbaum discovered meningococcus. Kitasato discovered tetanus bacillus. Olsen discovered plague. Next, we can see some important points regarding bacteria. So, bacteria are prokaryotes and unicellular, that is single cell organism. And another important point is that the 5 kingdom classification was introduced by R.H. Whittaker, and 5 kingdom classification includes Monera, Protista, Fungi, Plantae and Animalia. Bacteria comes under Monera, that is unicellular prokaryotes. Prokaryotes does not have any nucleus and other membrane bound organelles and they are unicellular, whereas eukaryotes contain nucleus and membrane bound organelles. They can be either unicellular or multicellular. Single-celled organisms of the domains bacteria and archaea are classified as prokaryotes, whereas in eukaryotes it includes animal cells, plant cells, fungi and protists. This diagram shows the differences between the prokaryotic and eukaryotic cells. Prokaryotic cells are small and simple, whereas eukaryotic cells are large and complex, and the size of the prokaryotic cells is 0.1 to 5 micrometer in size, whereas size of the eukaryotic cells are 10 to 100 micrometer in size, which is important. And prokaryotic cells are unicellular, where eukaryotic cells it can be either unicellular or multicellular. In prokaryotic cells, nucleus is absent, whereas in eukaryotic cells, nucleus is present. And prokaryotic cells has a circular DNA and eukaryotic cells have linear DNA. And prokaryotic cells have single haploid chromosome, whereas in eukaryotic cells they have paired diploid chromosome. Prokaryotic cells lack membrane-bound organelles. Eukaryotic cells has membrane-bound organelles. Prokaryotic cells reproduce both sexually and asexually. Eukaryotic cells mostly reproduce sexually. Prokaryotic cells in this cell division is by binary fission, whereas in eukaryotic cell division is by mitosis. And examples for prokaryotic cells are bacteria and archaea cells, and examples for eukaryotic cells are plant and animal cells including humans. Bacteria derived from the word bacteria meaning cane or rod. Anthony van Leeuwenhoek first observed the microbes under microscope and he named it as animalcules. And the study about the bacteria is called bacteriology. Father of bacteriology is Louis Pasteur. Study about harmful effect of bacteria on human body is medical bacteriology. Coming to the characteristics of bacteria, the size of the bacteria is 0.5 to 5 micrometer. It has been asked in so many nursing exams. The size of the bacteria is 0.5 to 5 micrometer. And bacteria are prokaryotic microbes and they have single chromosome and they are enclosed in rigid cell wall made up of peptidoglycan. So cell wall is made up of peptidoglycan. And in some bacteria, along with plasma membrane, it contains an additional membrane called capsule. And bacteria reproduce by mitosis and many bacteria form spores. Based on the nutrition source, bacteria can be divided into saprobes, parasites, photoautotroph and chemoautotroph. So saprobes are those microbes which feed on dead organic matter, and parasites are those microbes which feed on a host cell. Photoautotrophs are the microbes which uses sunlight to make food, and chemoautotrophs oxidize organic matter such as iron or sulfur to make food. So in next video we can see the structure of bacteria and classification of the bacteria and fungi and protozoa in detail. So thank you for watching this video. Bacillus.